Hi everyone, my name is Benjamin Machonti. So today I'll be taking you through biomarkers and proxies through this topic. Biomarkers and proxies as drivers of indirect insight from living systems to the cosmos. So before we dive in, I just want to bring it also to your attention that the terms biomarkers and proxies are used interchangeably throughout the video depending on the scene or instance being described. So without any further ado, let's dive in and see how these terms are applied in a wide variety of scientific domains. Both proxies and biomarkers serve as indicators and stands in, but they function differently. Proxies are indirect measures used to represent variables that cannot be directly observed and can be applied across fields like health science, economics or environmental science, and many more. Biomarkers, on the other hand, are specifically biological indicators that provide measurable insight into physiological states, typically derived from tissues, cells, or fluid like blood or urine. In medicine, biomarkers are crucial for diagnosis and treatment. For example, liver damage is often assessed using blood tests that measure enzyme levels. High levels of enzymes like aspartate aminotransferase AST and alanine aminotransferase ALT levels in the blood serve as biomarkers for liver health, indicating potential damage even before symptoms appear. This is because when the liver cells are damaged, these enzymes leak into the bloodstream, thus allowing healthcare providers to assess liver health without invasive procedures. Moving on to wastewater-based epidemiology as a proxy. Wastewater-based epidemiology is a technique that involves analyzing wastewater from communities to detect biomarkers, pathogens, or substances, providing a snapshot of public health within that population. It works by detecting traces of viruses, bacteria, or other biological markers that are excreted by humans into wastewater systems. By testing wastewater, scientists can probe the use of drugs and also identify the presence of infectious microorganisms in a community even before individuals begin showing symptoms, thus providing an early warning system for outbreaks. As such, wastewater-based epidemiology can serve as a proxy for population health in this case. Moving on to single nucleotide polymorphisms, which are SNPs. SNPs are single base pair changes or variations that occur at specific positions in the DNA sequence. This variation can be found in
animal DNA, plant DNA, all DNA.
SNPs act as proxies for genetic predisposition to diseases by serving as markers of genetic variation. While SNPs themselves may not directly cause disease, they are often found in regions of the genome associated with specific conditions. SNPs are often inherited along with specific genes or genetic regions due to a phenomenon called linkage disequilibrium. This means that certain SNPs are physically close to a gene on the chromosome and tend to be inherited together with that gene. For instance, SNPs near the BRCA1, breast cancer 1, and BRCA2, which is breast cancer 2 genes, are used as proxies for assessing breast cancer risk. These genes are generally known as tumor suppressor genes that play a critical role in maintaining genomic stability by repairing damaged DNA. Even though these SNPs might not directly cause disease, their presence indicates a higher likelihood of harboring a harmful mutation within the gene. Moving on to amino acid racemization as a proxy for fossil age. Amino acid racemization is a process in which amino acids within fossilized organic matter convert from their biological active form, L enantiomers, to their mirror image, D enantiomers, over time. In living organisms, proteins are composed almost exclusively of L amino acids. Upon that, the production of new amino acids stops, and L amino acids begin converting to D amino acids. This conversion happens slowly at a measurable rate. Over time, the ratio of L amino acids to D amino acids approaches equilibrium. The D and L ratio, the ratio of D enantiomers to L enantiomers, provide an estimate of how much time has passed since the organism's death. The older the fossil, the higher the proportion of D amino acids. Moving on to the cosmos. The Large Hadron Collider as a proxy for conditions after the Big Bang. The Large Hadron Collider, which is the LHC, is the world's most powerful particle accelerator located near Geneva, Switzerland, and operated by CERN. The Large Hadron Collider is housed in a 27-kilometer ring-shaped tunnel buried about 100 meters underground, spanning the border between Switzerland and France. At the heart of the Large Hadron Collider's operation is its ability to accelerate particles, usually protons or lead ions, to nearly the speed of light. This is achieved through a series of powerful superconducting magnets, which are chilled using liquid helium at temperatures close to minus 271.3 degrees Celsius. These magnets create strong magnetic fields that steer and focus the particle beams, while a sophisticated network of radio-frequency cavities generates electric field to accelerate the particles. The particles race around the tunnel in opposite directions and collide at specific points, reaching temperatures over 100,000 times hotter than the sun's core. These collisions produce quark-gluon plasma, a state of matter that existed microseconds after the Big Bang. Quarks are the fundamental building blocks of protons and neutrons, while gluons are the particles that mediate the strong force binding quarks together. In the early universe, quarks and gluons existed freely in a superhot plasma before cooling and combining to form protons and neutrons, and eventually the atomic nuclei that makes up all matter. By recreating these extreme conditions, the Large Hadron Collider acts as a proxy for the early universe, enabling scientists to study how it evolved from a hot, dense state to its current structure. These are some of the biomarkers and proxies used in the scientific world. So now, do you think you can identify potential biomarkers?